So we've now talked about the purpose of the bases of DNA and RNA, A, T, G, C, and U. And we said that they come in pairs because this leads to stability of the DNA. And this stability helps DNA from breaking down and helps you keep your DNA throughout your entire life and helps you preserve your DNA into your children. Okay, but that's not the only piece of the puzzle. That was only one portion of the DNA molecule. We've not taken a look at the entire DNA molecule yet. What we've only taken a look at are the bases that make up the DNA. And now we need to go to the second part. And that second part is our sugar. And we've already said that the sugar is pentose. So it's a 5-carbon chain. And if you look on the slide, there's your 5-carbon chain. So we've talked about sugars. We've talked about sugars making rings. We've talked about pentose. We've talked about hexose. We've talked about aldoses. We've talked about ketoses. And this is a pentose sugar. It is a aldose that we are talking about as well. So this 5-membered ring, well, here's my first carbon, there's my second carbon, here's my third carbon, there's my fourth carbon, and right up there's my 5th carbon. And this makes a ring just like we learned in our first course module in the biochemistry class. And then off of each one of these is typically an OH group. Okay, but we do have a modification of the sugar. What sugar is actually used for this purpose? Okay, well that's the question of the day, isn't it? We need to know what sugar is used. And the sugar that we use is ribose. So if you go back to that chart of the sugars, ribose is one of those structures I told you to memorize, like I did with glucose. And I told you ribose is going to come back around, and we need to know the structure of ribose now, and that way when we get to ribose later on, you won't have to memorize that. So ribose you should already have down pat. This is a couple of ways that we can represent ribose: in the straight chain ribose structure or in the ring structure of ribose. The carbons all have OH groups on them. That's the definition of a sugar, right? It had multiple OHs, polyhydroxy. And we see an aldehyde, a C double bond O at the end of the chain. And then this thing can wrap around on itself, and it can make this ring structure that's down here at the bottom. And this ring structure, well, it has an OH group at every single carbon. And this is my oxygen, my OH group that was at carbon number 5 that does the hookup for the ring. And then this is carbon number 4. This is carbon number 5 that has the OH group that's hanging off here. Okay, so what happens is that, again, we have an issue where we make something and Mother Nature uses something, and then Mother Nature needs to preserve it, and she doesn't want these things to become reactive. So just like with thymine and uracil, a modification is made. And a modification here is an OH group. And what we will see is that RNA, the R in RNA stands for ribo, and ribo represents the sugar that makes that structure up. Ribonucleic acid. So it uses ribose to do its purpose. Mother Nature takes the OH group off of a ribose. Which one? It's at my carbon number 2. And at carbon number 2, the OH group is removed, completely taken away. And if we do that, then we can help prevent some of these other reactions that take place with RNA. And if we remove those reactions, DNA stays behind a little bit better. So DNA stands for deoxyribonucleic acid. DNA, deoxyribo. D means minus, Oxy means OH. Ribo means ribose. So we're removing an OH group from the ribose structure. So if you go back and take a look at this picture, you probably question in the beginning, wait a minute, this picture, there must be a mistake. Because for a sugar to happen, well, this needs to be an OH group here, or here at least. And you didn't write an OH group on every single carbon like we learned back in the day. And you know what? It was here. But we're talking about DNA. And DNA is 2-deoxyribo. So that means this is carbon number 1 and this is carbon number 2. And on carbon number 2, I need to remove the OH group. And that's what DNA uses. A ribose that on the second position has removed an OH group. And that helps again with stability of the structure. So 2-deoxyribonucleic acid and ribonucleic acid. That are our two choices, and that's what you're going to see on this slide. So DNA and RNA, that's what those acronyms stand for. That's what those letters stand for. 2-deoxyribose and ribose. Those are the two sugars that get used in this genetic carrier. 2-deoxyribose for DNA only, and ribose for RNA only, and that's actually embedded in the name. So you know that's going to be one of the questions on the test. What does DNA stand for? What does RNA stand for? DNA stands for 2-deoxyribose nucleic acid, and RNA stands for ribose nucleic acid. So hopefully you will be able to answer those questions at least and score at least a few points on the test without being a major failure. Okay, so those are the sugars that we will talk about that DNA and RNA has in the actual structure itself. So here's a better view of a deoxyribose. It is ribose with, at the 2 position, the OH group is removed. This is the sugar that's used in DNA. If the OH group was on the number 2 spot, that's the sugar that's used in RNA. Okay, so what's next as far as the structure goes? Well, we've also mentioned this thing of phosphate. This is how they labeled the phosphate back in the studies to determine if it was really DNA that was getting transferred over or sugar that was getting transferred over, right? It was that phosphorus and sulfur that we focused on. So this phosphorus is going to be the link. It's kind of the thing that holds all of these individual pieces together. So we do have phosphate groups that begin to get involved in the structure, and that's PO4. Notice there's R1, R2, and R3 here because those oxygens can have other things attached to them, and then that phosphorus violates the octet rule. It's okay though, it's a double bond O, and then it's attached to 3 other oxygens. So there's 5 bonds in total. You know, octet rules really stay true to the elements up on the top of the periodic table, and the further you go down, the larger these atoms become, the more liable they are to violate the octet, and that's exactly what we have happen here. Okay, so finally, we put all the pieces together, and this is what we call the nucleotide. And the nucleotide is basically all 3 components hooked up. So up here at the top, we see the base, and then that's connected. I know this is a jagged little line here, but you just got to ignore it. But that's connected to the sugar that's down here at the bottom. And then instead of a CH2OH that's here off of the sugar, this is a CH2O, and that's where the phosphate comes in. So these are O minuses that are here on the ends. This is what we call the nucleotide. So we have a purine, we have a sugar, and we have a phosphate group. What purine do we have? Well, it looks like it's the one that has the NH2 group on it only, and that is adenine. So this is adenine here, 5-membered ring. It connects to the sugar, and the sugar is then connected to the phosphate. So that is the structure of the nucleotides that begin to hook up to make the deoxyribonucleic acid or the ribonucleic acid. So you got to pay attention. We first have a base, purine and pyrimidine. We then can talk about the nucleoside. So that's the bases and the sugar. We then can talk about the nucleotide, which is the base, the sugar, and the phosphate. And we have abbreviations for these. These nucleotides, because I don't want to write them out every time. So the proper names for those would be adenosine monophosphate. Okay, so this would be adenosine monophosphate. It's the adenine up on top, and then there's one phosphate group that's attached to that structure. So hence adenosine monophosphate. If that was guanine, guanosine monophosphate. If that was cytosine, well, that's again same kind of procedure all over again. You put monophosphate there. If there were two phosphate groups, adenosine diphosphate. If that was three phosphate groups, it would be called triphosphate. And they have abbreviations that go with them. So adenosine monophosphate would be called AMP. Adenosine diphosphate would be called ADP. And adenosine triphosphate would be called ATP. And ATP is very important because ATP is your energy. So if you take a biology class and you learn about metabolism, you will talk about the ATP molecule all the time. And ATP is a nucleotide. And ATP is made up of adenine and the ribose and three phosphates hooked on one by one by one by one, just like a train. One phosphate, two phosphate, three phosphate, go. Okay, but they all have abbreviations, and that's what those abbreviations mean. AMP, GMP, CMP, UMP. Sometimes we put the D out in front. The D just basically means deoxy, and deoxy is kind of the symbol that it's coming from DNA. So a lot of times if I put the D out in front, that really confirms to me that this structure is going to be found in a DNA molecule and not an RNA molecule. So it's kind of unnecessary, especially when I put T there, because I should know that thymine really only comes from the DNA molecule itself. But sometimes people like to be over picky and they put the D out in front. There's no harm in it, but it's there. So that's just some terminology that we can use for our nucleotides. So AMP, GMP, you got it. Notice GMP is not good manufacturing practices here. It is guanosine monophosphate. All right. So here's a slide that shows the nucleosides. Nucleoside is just the base and the sugar only. Notice there's no phosphate groups on these, so that's what we call nucleoside. And there's all kinds of nucleosides. Here's the adenosine and the guanosine and the cytidine and the uridine. Notice the ending changes on them. It's not adenine, guanine, cytosine, uracil, thymine anymore. So they change them slightly, so they put the side in it and you kind of see the scene at the end. Here are the structures involved in the RNA molecule. I know that because there's an OH group that's here on the sugars. And then not only that, but this is a uracil up here at the top, and that doesn't happen in DNA. Down here at the bottom, these are the DNA counterparts. So I know that these are DNA because this is second carbon, so this is 2-deoxy. So no H group, no OH group, no OH group, no OH group. And I also know that this set goes along with DNA because this is my T form. This is my thymine here. So thymine, and this is uracil. So uracil, I put a methyl group on it, change it to thymine. This piece becomes a little bit more stable now, and it prevents some reactions from happening. The same thing happens down here at carbon number 2. I remove that OH group, and I prevent some reactions to happen in this ribose structure. So that is the purpose of the nucleosides. Okay, so with that said, again, We can flip through the structures and talk about mono, tri, and diphosphates, but now we start to see the relationships with each other. We start to see how these pieces build on top of each other, and we start to see how these things make this DNA backbone that we will begin to call it. So we're still talking about DNA structure, and the next video will continue on with that.